really excited to join you here for the third series of the Aging webinar series. Today we have Melike coming, so really excited to hear from you. And before we start, just want to remind everybody about the Cambridge University Longevity Society. So we are a society of basically scientists and entrepreneurs, but also people interested in public policy. And so basically trying to bring everybody interested in aging research together. Just one note, we will have elections coming up, so anybody interested in joining the committee for the next year, reach out to Facebook or the website for that. So today we have Melike Dönertaş joining us, so welcome here. Melike is an EMBO postdoctoral fellow at the Leibniz Institute of Aging in Jena in Germany. And she started her scientific journey with a bachelor's degree in the Middle East Technical University in Turkey, with a degree in molecular biology and genetics. And the same institute she then continued with a master's degree. And then she came here to Cambridge to do her PhD at the EMBO, European Bioinformatics Institute. And this is where we've actually met at Audrey de Grey's talk here, one of these evenings. So Melike then continued her journey in Max Planck Institute of Aging in Cologne in Germany, where she joined the Valenzano lab. And then I think your lab moved to Jena in Germany at the Leibniz Institute of Aging, where now, as I've heard 4 months ago, you also received the EMBO fellowship to study the interactions of microbiome and aging, which is super exciting. So great example for us to see how a Cambridge graduate can make it in the world of aging research, and certainly great inspiration. So it would be very exciting. If you have any other questions also related to Melike's scientific journey, please go ahead, just write everything in the chat. And then at the end, we will just ask you to unmute and ask your questions. So yes, without, Melike's talk today is titled Intertissue Convergence of Gene Expression During Aging. So I think this builds on your PhD work. I'm sure you'll tell us more about that now. So without further ado, Melike, welcome here.
Hey, thank you very much. Thank you very much for the invitation and also this nice introduction. I'm very excited. I mean, I'm actually quite disappointed that I haven't met you guys when I was in Cambridge, but I had to do this in kind of that virtual setup. But so today, basically, my talk is actually quite short and it's very like biology or more like computational biology focused. But then I thought, I mean, we can still have like, I thought this would be a small meeting anyway, so we can have like a discussion and you can ask your questions and we can discuss. I would really enjoy more like interactive setup, to be honest. So I will just share my screen and all right. So I guess you should be able to see, right?
Yes, everything seems fine.
All right.
Brilliant.
Great. So basically, the title of the talk is Intertissue Convergence of Gene Expression During Aging and Loss of Tissue Identity. And before starting to talk about like specifics, I just wanted to also introduce myself, not career aspects, but more like in terms of science, what I'm interested in, what I work on. So basically, so I'm a computational biologist, and there are like different teams that I've worked on so far, and I'm focusing on a new direction now. Like historically, I worked on more like biology of aging, which involved both like more theoretical work, focusing on evolutionary theories, but also in collaboration with wet lab scientists. We also worked on regulation of lifespan using different interventions, different drugs. And then this was also kind of tied to my PhD work, which focused on drug repurposing for aging, where we used transcriptome or like protein structure-based drug repurposing studies to find whether we can reverse aging signatures with a specific focus on humans. And then another focus of my PhD was age-related diseases, basically where I was asking whether age-related diseases have common components that we can target to, target multiple diseases simultaneously so that we would extend healthy lifespan. Today, actually, I will talk more about this age-related deregulation of gene expression and overall like system-level disruption of gene expression kind of view. And this actually builds on my master's projects. And I continue to work on this during my PhD as well. And I will give more details in a minute. And actually now with my, my new direction is actually more focusing on aging and microbiome and focusing on both like microbial evolution, microbial ecology, and how gut microbiome changes during aging. Like I'm kind of, I guess, getting away from more like these translational aspects of drugs and everything, but more focusing on what happens in the microbiome and trying to understand its interaction with the immune system from more like biology point of view. But as I said, at the moment, now I will mostly talk about age-related deregulation of gene expression. And so overall theme in my work is kind of like making a distinction between increase in age and aging period, because I mean, in general, when you look at the definition of aging, we see this decline in functionality, susceptibility to pathologies, increase in mortality risk, and obviously this doesn't start from the first day in life, and we actually have more like this postnatal development where everything works fine, and then we have the aging period. And a general theme that I had was this comparative analysis of these two periods. And this actually comes from also these like theories of aging, where there is this developmental theory or hyperfunction theory of aging, for example, at the phenotypic level, which suggests aging is continuation of developmental program, and this is actually detrimental for the organism. Maybe this shouldn't happen. And then the other aspect is, on the contrary, maybe aging, the detrimental effects we see during aging is actually due to disruption of the developmental program or even reversal of it. And these are mostly linked to the damage-based theories, I would say. But one of the main questions I had was what happens at the gene expression level? Okay, maybe at the phenotypic level, there are supports for both of these theories, but in general, if we were to look at how genes activity change throughout, with time throughout development and aging, whether we see certain these type of associations. And this, as I said, like was something that I worked on during my master's actually years ago. The main question is whether aging is continuation or reversal of the developmental program. And this comes from both like evolutionary theories. You would think that like after the age of sexual reproduction, maybe the mutations that show their effect after the age of sexual reproduction, maybe they accumulate in the genome and they create a new kind of gene expression program. The other possibility is more like these accumulation of somatic or directly genetic mutations that can cause a change in the gene expression. And basically the question I asked was whether we see this kind of patterns, like during the development until age of 20, for example, in humans, whether we see a certain type of gene expression trajectory and whether it is reversed during aging or whether it continues. And in order to answer that, as I said, like I always worked in dry lab until this stage of my career, and I mostly use publicly available data sets. So I went to gene expression database, and I collected all the data I could find for human brain. And I specifically searched for these data sets that cover both developmental period and aging period. And basically, I just grouped genes based on whether like they go up, up, or up, down, down, up, or down, down. And what we saw was actually, I mean, if aging was continuation, if the gene expression program during aging was a continuation of the developmental program, we would expect like more of the continuation. But actually, we found like 50% of the genes actually reverse their expression program. This is like 50% you would expect by chance. So I cannot say that reversal is prevalent. But still, it suggests that maybe like aging is not the continuation of the developmental program. And more interestingly, since we have multiple data sets from different brain regions, what I searched for is whether the genes that show this reversal program, reversal kind of trend was common. And I found not the down-up, but this up-down pattern was shared across different data sets and brain regions. And these genes were mostly related to macromolecular modification, signaling pathways, neuronal synaptic functions. And since this is brain data, I at the time speculated that this is basically an indication of cellular identity loss, which was completely speculation at the time. But then still, we wanted to understand whether this happens in other tissues. Is it human specific? Do we see it in other animals as well, other species as well? And then this kind of led to a new direction in our research. And now I will be talking about this. So basically, this paper just came out like 3 months ago. And kind of like starting from this idea of reversal, we started to discover something interesting. All right. So basically, we teamed up with an experimental lab, Filip Kajtovic's lab, and we generated gene expression data from 4 tissues in the mice. Again, covering both like developmental period and also aging. And then together with a computational PhD student, Hamid, we started to analyze this data with a more focus on these reversal patterns. So first of all, when we just look at the gene expression, we see that during both development and aging, different tissues share certain developmental signatures or aging signatures. But then our question is more like about reversal, right? So then we started by just looking at the whole genome and seeing whether the changes in gene expression, whether changes in gene expression in the same tissue correlate or not. And basically, when we look at, for example, like between cortex developmental, cortex brain aging period, there is a positive correlation, but then for liver, for lung, for muscle, we kind of saw this negative correlation, which indicates a reversal pattern, even at the whole transcriptome level. And again, like quantifying how much of the genes show this reversal pattern, we again saw like around 50% across all tissues. And when we check the biological functions associated with them, for brain, for example, we saw synaptic signaling. For lung, we saw tube development, tissue morphogenesis, liver, protein catabolism, and muscle, cellular respiration, which kind of even supported our first speculation that this might be related to this cellular identity loss. And then related to that, we checked whether we see a sharedness between different tissues, because when we just looked at brain and different brain regions, we actually saw a prevalent, like a consistent up-down pattern. Do we see this across different tissues? And the answer was no. Actually, the gene expression reversals seem to be more tissue specific. So the genes that show reversal pattern in synaptic signaling does not overlap with the genes in liver, which were associated with protein catabolism. And around that time, actually, when we were doing this analysis, there was a paper looking at just the developmental trajectories across different species. This doesn't have any data on aging, but this is basically different colors show different tissues, and the shapes are different animals, and then this is a PCA plot. And as you can see, basically, it's kind of like early in the life. They actually almost like the tissues start from a common ground and then they start to diverge. And this was just a comment in the paper because it's more like evolutionary biology compared to transcriptomics kind of paper. But then this kind of gave me the idea, maybe if this reversal pattern is indeed related to tissue identity loss, and if this was already reported during development that tissues diverge from each other, I thought maybe with age, during old age, maybe they come back together and they start to converge. So I wanted to test specifically this hypothesis in this data. All right, and basically, I kind of repeated what they've done at the PCA space in the first instance. But by the way, I always say like but actually this is like a huge team effort, and Hamid was the lead person doing the analysis. I should emphasize that. But then, so basically, when we look at the PCA space, what we do is for each individual, we have 4 points, right? So basically, we just calculate the Euclidean distance between these points. And then what we saw is just like the paper published before, during developmental period, we saw that the tissues from the same individual start to diverge from each other. And during the young adulthood, actually, they reach to this top point where they are the most distinct. And instead, when we look at the aging period, we see this like tendency to converge because, I mean, it's not as strong as the developmental trend, obviously, but we started to see this convergence pattern. And this is at the PCA level, right? But still, like what happens at the gene level, which genes show this, this divergence convergence pattern. So basically, so here, what I'm showing is just gene expression pattern for one gene. Different colors show different tissues. And for example, this gene shows like a slight decrease in the muscle during development. It goes more or less the same during aging, but in the lung, it starts high, and then during aging, it starts to get lower. And basically, I thought, let's calculate this coefficient of variation, which should tell us whether these points are basically it just gives the idea about the variation, considering the mean expression level. But then it allows us to see how it changes with age, which kind of gives us an idea about this divergence convergence pattern. So for example, for this gene, we see that it diverges during aging because during development, because it's high in the lung, but it starts to decline in muscle, and this becomes even more lung specific gene. And then we start to see a decline in divergence because then they start to converge. And we calculate this per gene. And during development, we saw that most of the genes actually show divergence pattern indeed, like this. In aging, this is the number of significant changes. Number of significant changes is significantly lower in aging. I should emphasize that. So this is like a weaker signal in aging. But still, we see that the prevalence of convergence pattern is much higher. And when we look at the difference between convergence divergence ratio, it was a very clear and significant trend that during development, we have more divergence, and aging, we have more convergence. And then, but still, this is an overall trend at the gene level, at the PCA level, but then our initial idea was kind of like linking this trend with the tissue specificity. We asked whether this is relevant at all. And in order to do that, we first calculate the tissue-specific genes, just based on the data again, instead of like, we don't really use like markers or anything, but just gathering the information from the data. And then once we gather these tissue-specific genes, we asked what happens to them during development. And for example, here, okay, this is not a very explanatory figure, I must say, but basically, if it is like above 0 during development, it means it increases. And then, yeah, below 0, it decreases. And then we have the pattern during aging. And what we see is those that increase during development, those up pattern, 89% of them starts to decline during aging, for example, in the lung. And those that are already downregulated during development, only 6% of them show this down-up pattern, but they mostly continue down-down trajectory. So overall, this is just for the lung, but except for liver, we actually saw this strong association between tissue specificity and up-down pattern across all other tissues. And well, this supports our idea that reversal is related to tissue identity loss, if you wish. I mean, when I say identity, by the way, this is still postnatal developmental period. It's not like tissue differentiation, I must emphasize that. But still, I mean, we see this reversal pattern. And also these tissue-specific genes was significantly associated with the genes that show divergence convergence pattern from this analysis. And then we thought still we are not really showing the direct link. So in order to do that, we then grouped genes based on their expression patterns, whether they show like a decrease, their expression patterns during aging. So for example, this gene is specific to muscle, let's say, because it's highest in the muscle. And then, but then the maximum change that we see during aging is actually in the lung, and it declines. So this decreases in its non-native tissue. However, this one is more specific to liver, but then it starts to increase during aging in the lung. So lung becomes more like it starts to explain this this gene. This one is like showing a decline in its native tissue, and this one is showing an increase in its native tissue. And we thought this this scenario showing increase in non-native and decrease in native tissue would contribute to DiCo and tissue identity loss. So we asked which one of these scenarios we see in the data. And when we look at all genes, we already see that most of the genes show these two patterns. And when we especially focus on the divergent convergent genes, we see that a huge effect, and these two patterns were much more prevalent. This kind of suggested that indeed DiCo is related to tissue specificity and also related to the reversal pattern. But this was just one data set in mouse. The sample size is not huge. So what we wanted to do is we went to, again, public databases and tried to see if we see the same type of like convergence during aging in other data sets. And unfortunately, there are not many data sets that cover both developmental period and aging, so we cannot really study DiCo in other data sets, but we just focus on the convergence during aging. And basically, we use 3 data sets. One of them is microarray, quite old, 5 tissues, again, from mice. Another data set, which also has a single cell RNA component. And there we analyze both just these 4 tissues we had, but also with a larger analysis with 8 tissues, also again in mice. And then we also analyzed GTEC data, which is across humans. And again, we have enough data to analyze both just these 4 tissues, but also a larger tissue sample. And basically, I will not obviously go into the details of the table. What I want to say is, actually, I showed only like 2 layers of support for the convergence pattern during aging. One was this PCA-based changes, and the other one was this the proportion of the convergent divergent ratio during aging. And what we saw is, especially when we focus on the significant changes, all these data sets supported the convergence idea. At the PCA level, one of the data sets didn't show any support. But then when we focus on the link between convergence and tissue specificity, kind of like repeating this, this analysis, we saw that all these data sets, different tissues, different human, mice, they all supported the link between convergence and tissue specificity idea. And basically, lastly, we then asked whether the changes we see, this divergence convergence thing, or the tissue specificity, whether it is caused by the changes in cellular composition, because certain certain cell types become more or less prevalent, or is it cell autonomous? And we thought, well, this data set already has the single cell RNAseq data, then let's analyze it. So first of all, what we did is basically when you have a bulk RNA data, you can still deconvolute it to the estimated cell type proportions using single cell RNA-seq data, so that you would, in a way, you are saying how much of the expression variation in the data is explained by this type of cell or that type of cell. And based on this, what we focused on is, let's first just look at it here. We excluded all the genes that support our DiCo divergence convergence hypothesis. And then we looked at how the most, most prevalent cell types in each tissue changes with age. And we saw that actually, in general, during development, there is an increase, and during aging, there's a decrease. For example, neurons in cortex show this pattern, hepatocytes slightly, but we see this pattern, and this can already support the DiCo hypothesis. But then we asked, when we just focus on DiCo genes, do we see an even more prevalent pattern? Because if that was the case, we could say maybe that DiCo is mainly caused by the cellular composition changes. But actually, there was no significant difference between these 2 gene sets. And we said, okay, we cannot exclude the possibility that cellular composition contributes to DiCo, but at least it doesn't seem to be the only cause. So then we asked, we have the single cell data anyway. Why don't we analyze this pattern at the single cell level? So in order to do that, what we did is we took the maximally correlated cell types across tissues first. So these are, for example, endothelial cells across different tissues. And we asked how the correlation between these cells changes with age, during aging. And we see that actually there is a decreased similarity. So the same cell types become more divergent across tissues. And if we actually take minimally correlated cell types, so for example, these are from lung, it's club cell, brain, spiny neurons, they were the least correlated, or hepatocytes from liver and macrophages in the muscle. When we look at the correlations between these cell types, we see that there is an increased similarity during aging, suggesting these cells also, there's some cell autonomous changes that lead to these cells to become more similar, whereas, become more similar, that could have an effect at the bulk RNA level as well. So this is more or less the whole story, but then there are still many open questions, and I'm super excited to follow up on this study. So one of them is, for example, we are comparing postnatal development and aging, and I'm kind of making this huge leap to talk about tissue and cellular identity. One missing component is, of course, this cellular differentiation part. Yeah, at the moment we haven't looked at it, whether actually the postnatal development is kind of whether it is kind of the continuation of the like embryonic period, for example, or like the tissue differentiation. We don't know that yet. And also at the moment, we also don't know whether this has functional impact on aging. Okay, we see this phenomenon and we solve it across different data sets as well. But for example, if we ever manage to intervene in it, whether we would see a rescue of aging phenotype or whether this has any functional consequence at the moment, we also don't know that. And of course, we also don't know the mechanism, like whether this is caused mostly by, I don't know, changes in the gene expression program in general, somatic mutations or whatever. So basically at the moment, we also don't have data for that, unfortunately. And basically, this is what I wanted to share, and I should acknowledge both. So Mehmet Somen's lab is my master's lab, basically, and I collaborated with Hamid mainly on this project, and also Philip Kajtovic's lab. But then, of course, throughout years, development of my scientific program and everything got so much from Janet Thornton at EMBO BI, my PhD advisor, and also Linda Partridge, whom we collaborated very closely, and also my current lab, Dario Valensano's lab at FLI. So thank you for listening, and I'm looking forward to more like discussions and having a chat with you.
Great. Wow, that was super interesting talk. Are there any questions for Melike? Yeah, so first we have Lucas. Lucas, do you want to ask your question?
I think he can't speak right now.
Oh, can you not? Okay.
Yeah, you can ask.
Yeah, Lucas is asking, do you have any thoughts on the possibility of divergence convergence phenomenon occurring in the epigenome?
Yeah, that's a very interesting question, because actually during aging, we actually know that the variability between individuals, for example, increases during aging in the epigenome, like methylation levels. Between tissues, I don't think anybody looked at it, but actually there is like tons of data on methylome, right? I mean, it would be very interesting to see if we also see it at the epigenome level, and this could also help us to understand it mechanistically. And yeah, I haven't looked at it, but that's a good point. Actually, we can even start looking at it to start understanding the mechanism as well. Yeah.
Yeah. I mean, yeah, just a comment. I mean, it's like all that research on like hypomethylation, hypermethylation with aging. So yeah, is there a like with age, is there a general trend towards hyper or hypomethylation? Do you know? Or
I mean, I never worked on epigenome, so I shouldn't comment. I mean, I know that there is a certain trend, but I don't know which one. Maybe some of you guys know and contribute, but yeah.
Okay. So we also have another question from Iona. San, yeah, do you want to ask your question?
Yeah, as always, I have a bunch of questions. The first one is the following. So as you are presenting the data, I was trying to think, do you think is there evolutionary pressure for this reversal pattern, or your data simply suggests that, you know, we start from one single cell, and then as we develop, we go on different trajectories, and once that, you know, pressure is off because we don't reproduce anymore anything, that just simply brings the cells back to where they started from, in a way, is because you don't have any more the evolutionary pressure to keep cells well differentiated and each one have their own program.
I mean, I think I'm, I think more in the line of the second proposal, but then I think the lack of selection is still maybe not in terms of the differentiation of the cells, but maybe in general just maintenance of the gene expression program, like basically it could be even damage clearance, right? And it can still lead to disruption of the gene, because what we show is basically just the disruption. It's not really construction of a new genetic program, but it's more like it could even be achieved by things going wrong. So lack of pressure to continue damage clearance or something like that could lead to it. But yeah.
Yeah, and in terms of functional relevance, one of the areas that I was thinking is cancer, because I guess you know it's well known that during cancer development, some of the gene expression patterns may suggest some kind of reverse to the developmental stages. So I was wondering if you have any access to data sets where, for example, you look at pre-malignant biopsies and see if that shows an even more striking pattern of these gene expression changes.
Yeah, so actually at some point we did compare when we were just focusing on the reversal, not this DiCo pattern, but just the reversal, we checked whether the genes that we see this reversal pattern also show this like reversal pattern during cancer of the same tissue. Interestingly, I mean, we see a signal, but it wasn't more prevalent. So we were actually expecting to see something huge there, but it was like even less prevalent than the signal we saw in aging. Maybe one reason could be in aging, in cancer, maybe this reversal is more at the cellular level and more like differentiation kind of thing, and what we are gathering this signal from during development is more, I don't know, maybe inter-tissue organization or something like that. Basically, that's what kind of the reason why I want to look at more like embryonic development as well. And
I was thinking more in normal tissues near the cancer rather than in the cancer cells themselves, because I think the cancer cells are too complicated to understand.
That's a good point. Actually, we haven't looked at it, but it could be the case, like pre-cancer. Yeah, I think that could be the case. Yeah, thanks for the suggestion.
Cool. Stephen, do you also have a question to ask?
Yeah, that's really interesting. Thank you. I was wondering, I'm sure you're aware there's often really poor correlation between mRNA levels and protein levels. So I was wondering if you actually looked at the genes that are converging, whether they've been validated at the protein level.
That's a good point. Actually, we haven't, but then also with aging, the coupling between transcriptome and proteome actually gets disrupted as well. So I don't really have a strong feeling how that would behave, to be honest. But that's something that we definitely want to look at. Like, we weren't thinking about epigenome actually, but more like from transcriptome to proteome, whether the signal preserves. Yeah, we don't know yet.
And Xiaoming, go ahead and ask your question.
Hi, thank you. So I have two questions. First is that, have you looked at the overlaps in functions of those genes that are either converging or diverging during the process? Although they might be different genes in different tissue, but for instance, perhaps they're all involved in maintaining the structures of cells or functions specific to that particular organ. Have you done anything similar to that?
Yeah, so basically DiCo, divergence, convergence is across tissues phenotype, right? So that we cannot look at, but for the reversal, we looked at tissues, we looked at the genes in different tissues and their functions. And the most obvious signal was more like tissue-specific functions, but then one other function is more like immune-related stuff that comes up, which is kind of expected, like it's shared across different tissues, and we also see this this reversal pattern. But I don't remember. Well, okay. And regarding reversal, I don't remember anything about the like structure or like cellular organization kind of thing. But when we look at divergent convergent genes in general and their functional associations, we see all sorts of like extracellular matrix organization and like cell-cell adhesion and these kind of categories coming up.
Yeah, that kind of a follow-up question on that is that do you think, because when you observed some slightly different patterns across tissues, some seem more divergent or some seem more convergent, but do you think is it likely that they are actually the same process, but because certain cells, for example, epithelial cells, maybe because they are so homogeneous to begin with, compared to, for example, cells in the pancreas where there are more cell types, and sometimes they can have more phenotypic alterations to them during their lifetime. So because one group of cells are more alike each other to begin with, whereas another group are more divergent to begin with. That's why you're observing these kind of differences in convergence and divergence during aging, but they are all reflected by the same process of things like cytoskeletal deterioration and other stuff that you described. Do you think that's a likely hypothesis?
I mean, for reversal, it can happen, but then again, for DiCo, actually, we are comparing these same genes expression profiles across tissues, and then we see this divergent convergent pattern. Mm-hmm. So I don't think it can be the case. And also, like this, if I'm understanding the question correctly, to be honest, but and also at this cellular level, we also see this kind of relationship between different cell types across tissues. So even at the cell autonomous level, we see it. But I guess, I mean, still, I shouldn't exclude the possibility because just because we cannot check it or we did see support for the cell type specific thing doesn't mean we should exclude it.
Okay, thank you.
Thank you.
Yeah, Stephen, go ahead. You have another comment or question?
Yeah. Sorry, this was probably in the data somewhere, but I was wondering, you had mentioned convergence of different tissues of lung, liver, brain. Do tissues that compose of largely postmitotic tissues converge more than highly proliferative tissues? For example, the liver is obviously massively regenerative, and things like the gut and stomach will just proliferate continuously, whereas the brain is obviously almost entirely postmitotic.
Yeah, that's a good point, actually. So we haven't looked at it. So our choice of tissues was to actually sample all sorts of tissues as much as we could. So that's why we have muscle and brain, but also liver, for example. But then especially those data sets with like 20 tissues and in human GTEC data as well, actually we would have grouped the tissues and then do the analysis and then make a comparison whether the signal is stronger in one. And we haven't done it, actually. That's a interesting, yeah, that would be an interesting analysis to do, actually. Yeah. No, but we haven't done it, yeah.
So Brendan, sorry, I'll ask one more question too. Yeah, thanks for the talk, Melike. It was really exciting. I mean, I know you're focused mainly on the, I guess, the gene expression differences or the variability, but I was also looking at this generally, like what kinds of genes get upregulated. I know you said mainly it's the specializations genes, the cell-specific genes that seem to go down, or the variability seems to go down. But I guess in a way, Xiaoming already asked this question. I mean, is there anything in general that goes up? Because I found some conflicting data regarding to inflammation and sort of mitochondrial DNA damage. I mean, that's more like, I guess, total level of expression. But yeah, what's the current state of view on this?
Yeah, I mean, so specifically for the inflammation and immune related functions, for example, I think one big difference between different studies is the age range. And I see that when we just focus on, for example, in humans, the differences from age of 20 to 60, the changes we see is, I mean, contains the more like not that much healthy aging, right? But then as you start to include older and older individuals, these are most likely to be more like successful aging candidates. And then it's very natural that the overall trend changes. And in my experience, most of the conflicting data is actually because of these age ranges, because when I do the meta-analysis and when I just restrict the age range to a certain period and then analyze it again, I generally actually recapitulate most of the signatures in between data sets in terms of functions, not in terms of genes. That's very different. But in terms of functions, they generally actually overlap. Maybe that could be the reason why there is this kind of conflicting results, I could say, but yeah.
I see. All right, so data set choice is extremely important, yeah.
Yeah, like depending on what you want to look at, the age range, like even where most of the data comes from, with which age range, it affects everything, to be honest. And also even like doing either like group comparison or looking at continuous changes, because for example, I actually didn't really emphasize this during the talk, but we are always like looking at correlations, monotonic changes, and it is actually very different from looking at like young and old comparison, where you can really have like a huge change after certain age, and we are actually not detecting it. What we are looking at is more like continuous changes, and that could also change a lot.
Thank you.
Yeah, I also have a question. So you showed some graphs and you showed sort of like an up-down pattern. And yeah, so like what others have already said, there's the divergence that we initially see is probably, I'm assuming that's like increase in inflammation related genes. So as we age, I'm just wondering, so when you reach the maximum divergence, I'm assuming there's like a lot of like upregulation of inflammation related genes. Like does that stay high? And what are did you look at any gene pathways? And what, if you did, what were the gene pathways that like, so because gene expression started to come back down, or like or more back to baseline, to what your original baseline was.
Okay.
Yeah, so what were the genes that were sort of coming back down to baseline, or what were those gene pathways, or if you saw any?
Yeah. Okay. So first of all, regarding the inflammation or immune related stuff, again, like actually they don't really show that divergence pattern during development. We see convergence during aging, but actually, I mean, we also don't expect some tissues to have inflammation and all the others don't, I mean, unless there's an effect, right, in the physiological condition. So that divergence pattern is not that strong in inflammation related genes. But then during aging, actually they all start to go up, so they kind of converge. But then, so sorry, I forgot your like the follow-up part.
Yeah, I was curious which okay, so it's not really inflammation that's the one that's giving the biggest.
Inflammation shows reversal, right? So they generally go down and then they start to go up. But this is kind of shared between tissues, so they don't really show DiCo pattern that much. Yeah.
All right, thank you. So maybe just one more general question, Eddie and Melike. How did you first get into aging? And because I think was it in Cambridge or earlier? And then the second question, why microbiome now? I mean, what brought you to that?
Okay, so actually, so Yeah, I actually started my master's in comparative and evolutionary biology lab, and I got really interested in evolution of aging. And that's kind of how I was introduced to the topic. My first studies were mostly focusing on evolutionary theories of aging, whether we can test them using transcriptome data, genomic data. And then I moved to Cambridge, and where I did more like these drug repurposing studies and stuff like that. And then now microbiome is actually kind of combining all this aging idea and the evolution, because actually during the lifetime of an individual, the microbiome continues to evolve, and then there's the ecology part, the evolution part, and then the interaction between the hosts. So it's kind of like a combination of all my interests. So I guess that's why it's so tempting for me at the moment. Yeah.
That's very exciting. I wasn't aware of that. I think we have some other evolutionary biologists on the call, so maybe a similar interests. Yeah.
Yeah.
Great. Yeah, that was such an exciting talk, and that's why there's so many questions. Does anyone else want to ask any like final questions before? Okay. Yeah. Great. Melike, thank you so much. Sorry.
Just
Ionel, yeah.
Ionel, yeah.
Go ahead, please go ahead.
You tempted me. Yeah, based on this last thing that you just said about evolutionary perspectives. So do you plan to look at this sort of patterns in a wide range of species? Because, this mammalian thing may not be necessarily true for C. elegans or Drosophila or...
Yeah, definitely. I mean, so again, like I'm a computational biologist, I'm kind of like restricted by the available data. And unfortunately, most of the data from Drosophila or other species is more like not this like continuous range, but they really have these grouped comparisons with maximum of 3 time points or something like that. And it kind of makes it very difficult to look at this phenomenon. But I'm actually very tempted to try this. So actually my current lab is using killifish as the model species, and it's very short-lived, and we have like a wonderful animal facility here. So I'm actually very tempted to give it a go in different killifish species, and then see, because, okay, one more thing. So in killifish, actually, it's very interesting because developmental period is kind of like similar across species, but then the lifespan is very different. So that's kind of why it makes it like even more interesting to study more like with an evolutionary perspective. Yeah, I'm very tempted to do it actually.
Thanks.
Thank you.
Great, Melike, thank you so much for giving this talk. Yeah, anytime next time you're in Cambridge, let us know, and we can maybe like grab arrange a formal or something.
Yeah, and thank you for invitation. And I'm actually very sad that I didn't do this before the publication of the paper. You guys gave me so much ideas. Now I'm just very interested in just go and try them out. But thank you very much. This was really fun.
Thank you so much, Melike.
Thank you.
Thanks, everybody. Bye.